Hi, welcome to the miRspring Data Analysis Screencast. In this screencast, I will describe microRNA processing reports generated by the miRspring document. These reports are generated upon selecting the functional buttons at the top of the global visualization page. The three I'll be going through today are the export microRNA ID stats, the list seed abundance, and the microRNA cluster counts. But before I get to that, I'd like to quickly go through some microRNA processing terminology. I've recently prepared some slides on this, so I'll use one as a starting point. As you're aware, microRNAs are small non-coding RNAs, typically 22 nucleotides in length. They're known to bind to 3' UTRs, where they can cause RNA decay and or translational repression. And if we talk about the biogenesis, we know that these microRNAs are derived from longer stem loop precursors. So illustrated here is a typical stem loop precursor. Got the stem here, the loop, and microRNAs can be derived from either stem. If they're predominantly processed from the 5' arm of the stem, they're given a -5p in miRBase, and likewise if they're derived from the 3' arm, they've given a -3p in miRBase. So the miRspring document is heavily reliant on the miRBase entries. Ever since deep sequencing has been on the scene, we're aware that there has been a lot of variation that can occur at the ends of these processed microRNAs, these mature species, and they're the features that the miRspring document reports on. So the miRspring document can give you counts and statistics on the 5' isomiRs. Now just note that the default setting for the miRspring document is that a microRNA is considered to be a mature form if it starts within 7 nucleotides of that defined by miRBase. So that is any sequence that starts as defined by miRBase plus or minus 3 nucleotides, giving that 7 nucleotide window. The miRspring document can also give you statistics on the 3' isomiRs. It can also, so any tag that starts outside that 7 nucleotide window is defined as non-canonically processed, and the report will give you a count and number on that. The miRspring document can also give you an arm bias, that is, it will let you know what arm is preferentially processed from this precursor sequence. And I'll go over that a little bit more in the table. It can also give you a report on the microRNA length of the mature species, and it can also give you a report on mismatches, which you can infer to be RNA editing events. Okay, let's have a look at the report. Okay, so this report, that for all those features I just talked about, can be summarized through this export miRD stats, and this generates a nice table where the first column lists the precursor's ID as defined by miRBase and the total number of tags that are associated with that sequence. It'll give you the average length, so this is all these tags, it's the average length across those tags. And then the next 3 columns summarizes the 5p counts as defined by miRBase, or 3p counts, and anything that doesn't fall into those 2 baskets is classed as non-canonical. So just remember that in some cases miRBase may not have defined the 5p or 3p. Now this is a data set from the mouse, miRBase 19, and you'll see that most of the entries have a 5p and 3p number. But occasionally you'll see a dash, and this dash indicates that there's no entry in miRBase. So we can say here that miRBase 484 in miRBase 19 does not have a 3p coordinate, so therefore the miRspring document cannot give you a number. As we scroll across, the next column is the arm bias. So this is a ratio of 5p to the 5p plus 3p counts. So values closer to 100 represent that the majority of tags have been processed from the 5p arm, whereas values closer to 0 indicate that the majority of tags are processed from the 3p arm. The next 4 columns relate to the isomer counts. That's the 5' isomers and 3' isomers on the 5p or 3p tags. A little bit confusing, but just take your time reading that and it'll make sense. And that's listed as a percentage. The next 4 columns across are the absolute counts for the same columns. And then the last bit is editing. So absolute counts and as a percentage. Okay, so that's the that. The next thing I want to quickly go through is the seed abundance, and I've got another slide to help me work through this. And not there. Quickly bring it up on screen. Here we go. Okay, so we're aware that the seed sequence, which is bases 2 through to 8, are typically used by many to identify targets. I've identified that some isomers can have the same seed sequence as another family, and here you have an example here. So the LET7 typically starts with a U, but there is a small number of +1 isomers in data sets, and if you look at this isomer, it actually has the same seed sequence as the miR196A family. So therefore it can be useful in your analysis to break down the counts associated to a seed sequence rather than a miRBase name, and that's exactly what this reporting feature does. So I'll jump back to the document, click on the list seed abundance, and in here we can see a ranked list of seed sequences and the associated counts. Now the detail tabs in the last column will give you a readout of where all these counts came from. So for example, this value here, that's a bit busy. Let's go to okay. This one here, we can see that the seed sequence GTGCCGC in miRBase, this is derived from the miR293 3p arm, but from the data set that we've got here, that the seed sequence is coming from these microRNAs. Okay, the last thing I want to go through is the microRNA cluster account report. So as we are aware, microRNAs can be monocystronic, but they can also be clustered in the genome. Now, if they're clustered on the same strand, that could indicate that they could give rise to a polycystronic transcript, and this is indicated in the miRspring document with these histograms. We have the directional indicated by the text arrow, and the relative expression of these indicated by the height of the bar. In some cases, we can have clusters where they're on different strands, so it's not going to be likely that these are going to come from the same transcript. But all this information is in this last report I want to go through. So if we click on the miR cluster counts, we see a tabulated list. The table width will vary depending on how many clusters are at each coordinate. The first column defines the genetic coordinates of the cluster. The second column lists the precursor sequence separated by a space, and subsequent columns list the absolute value of those, so absolute counts for each of the family members. So in this top example, 12838 corresponds to the miR30A, and the 19179 corresponds to the miR30C. Alright, there we go. 3 simple reports that will give you a great way to start some downstream analysis, all in a nice tabulated format. That's all there is for this report, for this screencast. Thanks for listening.